Bom dia para todo mundo, buenos dias para todos, good morning to everyone. We have here
Good
people
morning.
from all over the world, right? So that can join us for the webinar, for our first webinar for Qsar. And first of all, I would like to thank all audience which are here with us. We have here toxicologists, we have here registration professionals, which are interested to know a little bit more about the Qsar services and Qsar information, which is important too now, and that will be more important in the near future. Right, so we have here 10 participants on the room right now, but we have 24 already registered, so they might come during our presentation, right? Okay, so how it is going to work. I will explain how this webinar will work, right? So I'm going to make some questions to Pedro, which is our partner in Argentina, which performs the Qsar services together with SmartTox, right? We are partners to provide the services to the market, right? And I will make like exchange questions to Pedro, which will explain a little bit more about certain points of the Qsar, which is important to you to understand what the Qsar is, what's the main targets to achieve in the Qsar services, right, in the Qsar studies, and also to explain how the expert system works, right, and each different special isomers that we have, right, and closing it, we are going to explain the existing data and the critical analysis that needs to be performed on this evaluation. Okay, we are going to have like 30 minutes of webinar, right, and the 10 remaining minutes we are going to keep for questions. So please send your questions through the message here for the Zoom platform, and we are going to gather all this information, all these questions, and in the remaining 10 minutes, we are going to answer all of them to you, okay? So I think I talked too much already, right? So about the introduction of the webinar. So let's start with Pedro. Pedro is a PhD in the chemistry, right? So he's responsible for gather all the information from the expert systems and make all the relation between the chemistry and the biology activity they found in the expert systems, right? So the word is all yours, Pedro. So introduce briefly yourself, and when you finish, I'm going to start with the first question to you. Okay?
Okay, thank you very much, Alexandri. I'm Pedro Gwizdalski. I'm a PhD in organic chemistry. I got my PhD at New York University in 1989, and since then I've been working especially in the veterinary sector and since 1994 in the agrochemical sector, doing process research and development, formulation development, synthesis, and now I'm concentrating into what is the data and the data that it needs to be generated in order to register a product in different countries. Chemistry, physical chemistry analysis, toxicology, ecotoxicology analysis. And since 10 years ago, we started with this platform, which is called Qsar. And we've been doing a lot of Qsar analysis, and we have a lot of expertise on that. And our experience tells us different ways to analyze Qsar analysis, which is very important for equivalency registration. based on FAO, obviously, on the guidelines of FAO.
Great. So, Pedro, tell us what exactly is the Qsar, right? I think everybody here, which is together with us, wants to know what is really is Qsar. What is this tool? What is this tool is meant for, right? And what's the importance of within this product for the equivalence analysis for the agrochemical products, which is intending to be registered in Brazil and in other countries in the world.
Okay, Qsar analysis is quantitative structure-activity relationship. So there is a relationship between the structure and the activity of a certain compound. And it's based that similar molecules behave similar. And based on FAO also, chemical products that are quite similar, same chemistry, same biology. And there is a little bit of history related to some relationships. For example, in the 1800s, in the mid 1800s, it was found that certain alcohols and its behavior and its biological activity was related to their solubility in water, for example, or their molecular weight, or some narcotic effects of some non-polar or non-ionizable compounds were related to the partition coefficients. Even in the 1960s was Hans tried to have a parabolic relationship between the partition coefficient and the biological activity for an analogous series of compounds. So that was the Hans analysis. We have to understand that when you have a new impurity in a matrix, in an active ingredient, that new impurity could generate certain biological activity which will compromise your active ingredient. Usually that impurity is either formed through side reactions or through some degradation of the active ingredients during the synthesis. So it will generate that impurity that is probably not in the reference. So in order not to isolate that impurity and run all the tests that you need to run, like physical chemical properties, like toxicology effects or toxicology studies or ecotoxicological studies, you have a tool that, once again, is the Qsar analysis. That Qsar analysis is based in homologous series, which are related both to three main points. One is there are groups alerts which categorize that impurity. Its similarity structure, and that will generate certain a database that will be correlated to the estimated physical chemical constants. In this case, the partition coefficients or lipophilicity. Okay, so that's why it's so important to have also a very good knowledge in chemistry, connectivity, groups alerts, mechanisms of actions. So basically that's what Qsar is. There are different platforms which they are constructed in different ways, but at the end they will be correlated to each of one because as many people know, most of the authorities ask for 3 different platforms, run the endpoints for different platforms. And those endpoints are very crucial. If you want, I could list those endpoints, but do you like me to list it, or the people that that are attending this webinar know already. But let me say, there are some endpoints that have a value, like LD50, like inhalation and continuous are you hearing me? Okay, and then you have some non-value like constants, like mutagenesis, carcinogenesis, teratogenesis, skin synthesization, eye irritation, and skin irritation. So those models, different models, those different platforms will give you different results, and that you will relate it. That those existing the data that you obtain, you will correlate to the impurity and to the active ingredient and homologous series. And those homologous series obviously are related to structure, to activity, to mode of action, and you will collect and you will select in some of the platforms are you are allowed to, and some other platforms you are not.
Great. So we know that the Qsar is requested by the authorities, Brazilian authorities, when it goes from the phase 1 to phase 2, in order to know a little bit the profile of the toxicology profile of this product before it goes to the toxicology studies itself, right? Mm-hmm. But Pedro, when we talk about data, right, the data mm-hmm. obtained for each platform, you retrieve mm-hmm. A lot of informations in the platforms. So when you retrieve these informations, each platform alone is sufficient to carry out a report and justify the results of each expert system. Like one of these expert systems, like the private one, will be enough to justify the report, to justify that toxicology evaluation. Why the critical analysis is so crucial for this Qsar evaluation when you do that retrieving of information from each platform?
Well, in our opinion, in order to have a robust report for Qsar, then the results that you obtain in each platform is not sufficient enough to do the report. Why we think that you have to have a critical analysis? That critical analysis is related not only to the results obtained, but also related to the structure itself. You have different groups within the molecule or that impurity that you are analyzing against the active ingredient and against structure that are similar. For example, you could have a chlorine, a bromine, an iodine, or other groups like double bonds or benzene rings that are substituted benzene rings. And for example, in a double bond, you could have attack of an oxygenation through cytochrome P450, which will oxygenate, make an epoxide, which is highly reactive, and that could react with the basis of the genes of the ADN, which are composed of nitrogen-related parts of the gene. So there you can have mutagenesis or carcinogenesis. So, but if that double bond is somehow electronically not possible to be attacked, or sterically that double bond cannot be attacked because you have very voluminous groups, even though you could get a positive result because of the platform, chemically some of those reactions are not possible. So that critical analysis is so important. So you need to know a little bit about chemistry, a little bit about the impact of those groups, the impact of those groups in terms of mechanisms of action and organic chemical reaction and their mechanisms. For example, when you have a chlorine, you can have an SN2 reaction or an SN1 reaction. But if that chlorine is not shielded enough, the reaction could not occur. So besides of obtaining the results that each platform gives you, you need to analyze why is that a result that you obtained. So besides enumerating all the results that you get from the different platforms, you need to have a critical analysis, and you need to know a lot about organic chemistry also. So you have a relationship between what the result is and what the logical of that result. So you can justify why that particular result that you get is reasonable or is not reasonable. So we think that just the results are not enough. And we try to analyze that in a critical way in order to have a very robust report.
Right. So that critical analysis is also together all the toxicology information together with the chemistry that you have retrieved from the system in order to create a well understanding of the hazard of the impact of this compound, right, chemically in to the authorities. Is that right?
Mm-hmm.
Okay.
Yes.
Right. So
that's the objective. So when the authority sees that report, sees that you have a very robust analysis on the result and the impact of those results and the justification, the chemical justification of those results. So there is a very important point here that you need to address in that report.
Right. So to everybody here might know that for Qsar evaluation, the platforms used can be as a public domain or as a private domain, right? So when to use those platforms in each authority, that depends on each country, right? For Brazilian registration, for example, you can use just the public domain platforms to retrieve those informations and make that correlation with the existing data and also the toxicology information available to create that knowledge, right? So for other countries like Europe, they request to use a private platform that's not well written anywhere, but for example, the OECD 69 describes some private platforms there, which is the Moodcase, Topcat, and also the Derrick platforms, which are more robust platforms to retrieve those informations, right? So as Pedro has explained, one platform is not enough to retrieve all this information, but a combine of these platforms can create a better scenario to explain the toxicology results retrieved from those platforms, right? Pedro, please.
Yes, continue.
Right. So in this scenario, we are talking about retrieving the information from the platforms, right? So we evaluate also the active ingredient and the impurities, and we make the comparison between them. So what the aspects should be taken into account when the structure, about the structure of each impurity, right? Concerning like the structure itself, the analogs, the geometric a special
Yes.
structure?
Well, when you have an impurity, as I said before, that impurity could be generated through different mechanisms. Some of those mechanisms usually are side reactions or degradation, or they come from the raw material that you have used in your process, and that impurity is generated. What you need to look at the impurities, first of all, what are the alerts, right, that you may have? Those alerts are related to groups, as I said before. For example, when you have groups that have toxicology impact, like N-nitroso or azoxy compounds or oxyazoxy compounds, or others that you already know that they have a toxicology impact. But when you have groups that are not listed as of toxicology impact, some of those groups could generate in your matrix a toxicology impact, and that's related to the obviously on the concentration that you have that impurity, but also the structure of that impurity, which is always sometimes is related to the active ingredient. So when you make that analogous series, you have to take into account all those things. So that's the importance of those analysis. The different platforms, as you said before, different countries tells you the different endpoints that you need to understand, that you need to report. And those endpoints, for example, in Mexico, they only ask you for LD50, the only endpoints. But for the others, for other authorities, you have, as you said before, private and non-private platforms. For example, OECD, which is one of the non-payable, non-private platforms, allows you to create your own series. That series, obviously, you're going to have to create in terms of the structure, which will be related to the active ingredient and to that impurity, okay? But others, like EPA, it will iterate its own series. So that's why you have to have different platforms in order to analyze the impact, the toxicology impact of that impurity. We need to understand that the impact of that impurity will always be related to the matrix. That is, that impurity in your active ingredient that is not in the reference, what will be the impact of that impurity? And as I said before, it's very well related to the structure. And when you make your analogous series, you need to relate to something that has the same mode of action. For example, if you have a neonicotinoid, obviously you're gonna select the neonicotinoids that are quite related to that impurity, okay? Even within neonicotinoids, you have the nitronicotinoids and the neonicotinoids. But in general, they have the same structure. They have a nicotinic structure or a nicotinic related structure. So that's why it's important to understand structure, chemical structure, but also the connectivity, the groups that you have, and that you will create the series. OECD allows you to do so, but other platforms do not. What is very important is the domain. That is, every time you go to to the different platforms, and you create without impurity a series of analogous compounds, that particular platform will have more database. So the more database you have, the better it is, because you're going to relate the properties, which is an algorithm that the platform creates, which will be related to the groups alert and with a similar structure and the similar mode of action. I don't know if I respond, I answer your question correctly, or there is any doubt.
That's just a discussion that we are going to have to press through all the information that we have for all our audience. But focus on what we just said. These impurities, they are very similar because they are impurities from the active ingredient. So they are very similar. And in cases that the expert system retrieves all the information, you have some structures which are different, especially there's a special isomer differences, right? So the expert systems considers that different special isomer like geometrical structure and enantiomer structures, because sometimes some of our clients request some evaluations, and we have just one impurity, but we find that this impurity has an enantiomer or has an analog which needs to be evaluated too. So explain a little bit better to us how it works, in order to our audience knows better why it's important to make this evaluation from an analog, an enantiomer, or a similar special geometric structure from each impurity.
Well, one of the fragilities of all those systems is that they don't discriminate between enantiomers. When you have an enantiomer, the expert system doesn't discriminate between one enantiomer or the other, or geometrical isomers too, whether they are cis, trans, and E or Z isomers. So the expert systems, all the expert systems, still today, and this is one, as I said before, this is one of the fragilities of these platforms, do not discriminate between enantiomers or stereoisomers, or, as I said before, geometrical isomers. But sometimes you may have an impurity that is not related to the active ingredient. So that's why I said before that it's important to understand the structure. So you will look for structures that are similar to that impurity, even if that impurity is not closely related to the active ingredient. So this may happen. It's not usual to happen because, as I said before, most of the impurities are quite related to the active ingredient through the composition or site reactions or reactions that are overreacting. For example, you may have a chlorination reaction and you may have one substitution of one chlorine or a substitution of two chlorines. So that could generate an impurity with two chlorines. So you will have an alert on that. Or a nitration reaction. You can have one nitrate, NO2 in the molecule, or you may have two nitro compounds in the molecule. What is very important also is to understand the mechanism of reactions. Why? Because you may have a polynitrated compound through nitration reactions in different positions, and those different positions could have different impact in the molecule. It could have the nitro groups attracts electrons. So if they attract electrons, it's very possible to have a base, a gene base to attack there, and you may have a carcinogenic impact. So the position at what that group is in the molecule is also important. So that's why I said the critical analysis is also very important. But what is also very important is that when you said that impurity is the one that you said, you have to elucidate that impurity and be sure that that impurity is really the one that you have, because that could have an impact on the Qsar analysis. The position at what that extra group, which is not in the reference, could have a really potential impact. For example, we had a case on fomesafen, and fomesafen you have a reaction, a last reaction, which is a nitration reaction, and you have two benzene rings. It's not the same that the dinitro compound could get into one benzene ring, then it could go to one benzene ring and the other one to the other benzene ring, because polynitrate compounds are extremely toxic. So if you don't elucidate that structure correctly, you could get a potential toxicology effect that really is not the one that you are expecting, because that impurity was wrongly elucidated. So that's why it's so important to have a critical analysis, also to understand the structure, relate that structure of that impurity with the mechanism of actions, with the structure itself, with the active ingredient, and with homologous compounds. Although today the authorities do not ask to relate that impurity structure with homologous compounds, and that homologous compounds usually may have the same, or should have in the best way, should have the same mode of action, or at least the same chemical structure. If it's imidazolidone, then you will relate to imidazolidones, and you will relate to imidazolidones that have the same mode of action. So you try to that homologous series fall into the domain that you are analyzing. You are muted.
Quezada, your microphone is off.
Oi, diga, Marcelo.
Now it's okay. Your microphone goes off. I was
okay.
mute.
I didn't notice that. So, and we are running out of time. We have like 2 minutes to end these 40 minutes that we have scheduled, right? So it's a lot of knowledge to share with our audience. But I will close with like a last question, which is like concerning to the evaluation of the authorities. Like all this retrieved information that we take in together with the existing data and the critical analysis creates a quality, a robustness and repeatability of the information which will be evaluated by the authorities. So
yes.
It's very crucial that can be repeatable by the authorities, and all the information is available to the evaluation, to the study. You know, once the authorities will follow the same path that you have shown in the study, but not only for that. They will create another scenarios because they have access to it, and you need to express all the results that you have, but you need to justify all of them. So once you have bad results, and the Qsar study, that doesn't mean that will not be acceptable by the authorities. Is that right, Pedro?
That is correct. You need to understand that some of the platforms are basically based on connectivity. So if you have a nitro compounds, they can have a connectivity to a nitroso compounds, and a nitroso compound is toxicologically relevant. So they will relate that series to also to nitroso compounds. So it will give you a bad result. But that's why the critical analysis is important. Why and the literature also is important but because you have literature on, especially on active ingredients, obviously they are all published in FAO, in EFSA, in EPA, but also not only that, but if the company that has generated the study do also have the data, the tox data, the 6-pack plus mutagenesis and Ames test, and all the data they may have, it could help also in the analysis, because you're going to relate everything to a matrix. The impact of that impurity in that matrix. Even you could calculate the impact it will have. FAO has a calculation of the impact in the concentration of that impurity in that particular matrix. So, but bibliography data and data that the company, the one that has that active ingredient with that particular impurity through a manufacturing process is also important in the analysis. Okay. So everything is related.
Okay. So Pedro, we have just 1 minute left. So I will open to questions to everybody who wants to question. I noticed that nobody write any questions here, but if you want to make any questions here during while you have just left 1 minute, please do it so. But afterwards, you can also write by LinkedIn or by email to us in order to make those questions. So it's open now who wants to make any questions.